Am I audible to everybody?
Yes, sir.
Okay, fine. So I got to know from Roy sir that he has taught you something about the basics of immunology, like innate and adaptive immunity. Is it right?
Yes, sir.
Okay, fine. So I was just going through your syllabus, and I just went through the first unit that talks about the basics of cancer biology. Right?
Yes, sir.
Okay, fine. So if we first start with the cancer and how the cancers result and what are the causes and how are the cancer cells special, I think that would be quite better for all of us to start with, and then we can move forward. So I first want to know about the background of all the people who are present over here. Hello?
Sir, some of us are from non-medical,
Like
like me, sir.
which subject?
Sir, physics, chemistry, and mathematics, nanotechnology.
Okay, okay. You're presently pursuing nanotechnology, masters in
Yeah.
nanotechnology,
Yes, sir. Yes, sir.
and others like others. Any other
Sir, zoology.
zoology, other than well, biological science or nanotechnology? Any other person? Anybody from biotechnology or biochemistry or clinical biochemistry? Sorry, biochemistry. Okay, okay, fine, fine, fine. So let's start with the cancer biology. If we talk about the cancers, so cancer is the abnormal proliferation and growth of cells anywhere in the body. Fine. So there is a difference between normal tissue and the cancer tissue, normal proliferation and the proliferation related to the cancers. Fine. So the cancers, in case of cancers, there occurs the abnormal proliferation, meaning to say, if in normal circumstances or in the normal tissues, there is the requirement for the growth of cells to a certain limit. For example, there is requirement of the cells to double to certain extent. But in case of cancer cells, they not only double, they quadruple, they become say 6 times, 10 times, 20 times, and so on and so forth. So they grow without any controls. So this proliferation results in the abnormal growth in a particular tissue. That abnormal growth, if it is localized to that tissue, if it remains there for longer period of time, or if it remains there for lifetime of a person or a patient, it's known as tumor. So unless and until it has these cells have left from their primary tissue to the other parts of the body, it's known as tumor. Are you getting me?
Yes, sir. Yes, sir.
Okay. So now if they move away from that primary tissue, for example, if this abnormal proliferation of cells occurs in the lung tissue, fine. If it remains localized to the lung for longer period of time, it's known as tumor. But now if it metastasizes or moves away from the lung and goes to the kidney, for example, or say for example the adrenal glands, it this tumor is now known as the metastatic tumor or cancer. Fine.
Yes, sir.
Okay, so at this point of time, we should be able to differentiate between cancer and a tumor. So a tumor is the abnormal proliferation of the cells localized to a particular organ. Now, if these cells move away from that organ to the other organ, this is now known as cancer or the metastatic tumor. Fine.
Yes, sir.
Okay. Now, there are types of cancers. One is known as carcinoma. So, carcinoma is the cancer of solid tissues or the epithelial tissues. The example of the epithelial tissue is, for example, lung, the colon, esophagus, so on and so forth. So, if the cancer or the abnormal proliferation occurs in these solid tissues or the epithelial tissues, it is known as carcinoma. Fine, it's known as carcinoma. Now, another category is the sarcoma. It's again the solid tumor, but it occurs in the tissues like bone and cartilage. So, it is known as sarcoma. So, in other words, we can say that the sarcomas are the tumors of the bone or the cartilage. Fine.
Yes, sir.
Am I audible to everybody?
Yes, sir. You are audible.
Okay, fine. Now there is another category of cancers. Those are known as, in layman terms or in the simple terms, we call those as the blood cancers. Fine. But the blood cancers in layman terms again has the two types. Scientifically, it has the two types, or technically, it has two types. One is known as leukemia, and another is known as the lymphoma. So we can differentiate between the leukemia and the lymphoma, but essentially they are the cancers of the blood cell types. I mean the cells which are present in the blood. For example, the B cells is known as B cell lymphoma. T cells is known as T cell lymphoma. If it occurs in the another cell types, it's known as leukemia. Fine. So we will as we move on in further lectures, we will get to know what is leukemia and what is lymphoma. What are the cell types that give rise to the leukemia, and what are the cell types which give rise to the lymphoma. We will talk about those in detail in another lecture. Now, if we talk about the genesis of cancers, how these cancers or how these tumors result. So for that, we first need to understand the simple biology of a tissue. For example, if we talk about the for example, lung tissue. If we talk about the lung tissue or any other tissue, for example, the skin tissue. So sort of, I will try to explain a different way. Suppose any person or anybody among you gets a wound, or he gets an insect bite. So what happens in that case is, if a person gets wound, that wound gradually heals up with the time. This is the normal way of talking, or this is the layman's way of interpreting the healing of the wound. But now, if you talk about technically, if you talk about the biology of the wound healing, what happens is that the tissue or the part of the body which gets a wound heals up because of the growth of the cells, because of the division of the cells. So the cells which have not been hurt or which have not been injured, those cells divide and cover up the tissue, or the cover up the part of the body which has the wound. So finally, what we can see, those cells dividing in a normal and controlled way, and finally and gradually they cover up the entire tissue. And at the end, say after 10 days or the 15 days, you can see that the wound has healed up. So this is the normal course of event that happens during the normal physiological process. Now let's compare this process and contrast it with the process of tumorigenesis, or process of the cancer formation or the tumor formation. Are you getting me?
Yes,
Yes,
sir.
sir.
Am I
Yes,
clear?
sir.
Am I clear? Any questions so far?
No questions.
Okay. I will move on. So now, if we compare the normal process of cell proliferation or cell division, which I gave you the example of wound healing, if we now compare it with the process of tumorigenesis, in case of tumors, what happens? The same cells For example, epithelial cells, they divide without any controls. For example, if under normal condition, a cell needs to divide up to the 3 generations, or if a cell needs to form 5 cells, this cell under the process of tumorigenesis does not form 5 cells, it forms 20 cells. Likewise, another neighbor of this cell again forms 20, another forms 30. So what happens? This results in the abnormal growth within an organ. I mean to say, a different kind of organ having abnormal physiology is formed within the same organ as a result of the abnormal proliferation or the abnormal growth of the cells, or some cells, or at least some cells in the tissue. So what has happened to those some cells which have resulted in the formation of the tumor? Why have they divided unruly? Why have they divided without any breaks? Why have they divided without any controls? So this is a big question mark. What is the answer? How do we answer this enigma? There are normal cells in the same tissue. There are abnormal cells in the same tissue. What is difference between those normal cells and what is the difference between those abnormal cells? Are we able to answer? If we are able to answer this question, this difference, surely we will be able to understand the process of carcinogenesis or the process of tumorigenesis. So the first answer, or the first explanation, or the primary explanation to this problem is that there occur changes in the genome or the DNA of those abnormal cells. So if I isolate the DNA or the genetic material from the normal cells in the same tissue and the abnormal cells in the same tissue, will I be able to will I be able to spot certain changes, certain abnormalities in the DNA of the abnormal cells as compared to the normal cells? Are you able to get me what I'm trying to say?
Yes, sir.
Okay, fine. So let's move on. Now, if I check the genome of these abnormal cells, I will find certain changes. What are those changes? So this is these change. This is the layman's language. Change. What do you mean by change in terms of a molecular biology perspective? So these changes are basically known as the mutations. Fine. So what are the mutations? Now, what is the term mutation? So most of you you must be well aware of what DNA is made of. So DNA is made up of nucleotides. We have adenine, guanine, cytosine, and thymine. So adenine pairs with the thymine, adenine pairs with thymine, and cytosine pairs with the guanine. Fine. So this sequence of these nucleotides, these base pairs, make the DNA. Now, if there occurs certain changes, for example, adenine, as I told you, adenine base pairs or adenine pairs with the thymine, and instead of the thymine, there comes cytosine. So this base change is known as mutation. And if this mutation results in the protein, because as we know, DNA encodes RNA, then that RNA is being translated to form protein. Now, if this change or this mutation has resulted in the protein which is abnormal, and now if this abnormal protein is very important in the cellular physiology, I mean to say, if this protein has been changed because of that mutation, it will now be detrimental to the cell. Because under normal condition, this protein has to perform a specific task. Now, it's not only performing that specific task, it's performing another task. Or if this protein under normal condition has to work up to certain limit, under mutated conditions, this protein is not only is working above that limit, above its requirements, which results in the change in the physiology of the cell, which results in the change in the function of a cell. As a result, because of this change, because of this change in the DNA, which gets reflected in the protein, and because of that change in that protein, the protein gets modified in a way that it performs an abnormal function, or it performs the function well above its limits. And entirely, if we put sum up this sequence, this change starting from DNA to the protein level, it results in the change in the function, it results in the change in the physiology of the cell. And ultimately, a cell which up to this point of time was behaving normally, it will now behave abnormally in a way that it will proliferate much more, it will divide much more. So this entire sequence of event culminates in the overgrowth, abnormal proliferation of those cells in the body, and finally a tumor results. Is this clear?
Yes, sir.
Any questions so far? Any questions? Any
No, sir.
questions? Am I going fast, or it's quite okay? You can
No,
tell
sir,
me.
it's okay.
It's okay. Okay, fine. So now, so this was something about the basics of cancer biology, how cancers, how cancers result, what are the changes that mediate the process of carcinogenesis. This was not though the entire story, but it's at least some basics. Now, let's talk about the hallmarks of cancer. How do we get to know that this is a cancer cell? How would a molecular biologist or a cancer biologist identify a cancerous cell or a tumorous cell? So there are certain hallmarks of cancer cells or the cancers which differentiate them from their normal counterparts. Fine. So these are few hallmarks which help us to differentiate between the normal cell and the cancer cell. And these hallmarks in fact are the capabilities, they represent or they depict the capabilities of these cancer cells by which they grow uncontrollably, they divide uncontrollably and form the complete tumors or the outgrowths within a organ or within an organ system. So the first hallmark of the cancers is the sustained proliferative signaling. Fine. What do you mean by the proliferative signaling? So let's first talk about the proliferative signaling or the proliferation of the normal tissues or the normal cells. So in case of normal cells, what happens? There is requirement of the division, as I in the beginning talked about the healing of the wounds, or if a certain part of the body, say a colon, is removed due to the surgery, so there should occur the growth of the colon, which will sort of cover up the lost tissue, or which will cover the wounded area. So where do these signals come from? So the cells, the normal cells, or which are present within the tissues, they produce certain signals, or their neighboring cells within the tissue produce certain signals which are known as the growth proteins. Once these proteins are secreted, once these proteins are present, the growth factors are present, they bind to the receptors on the cells and help in their normal proliferation and division. But this is entirely the controlled process. How it is the controlled process? Because these growth factors or proteins are produced in certain amounts or certain limits. After that, their production stops. So this is the normal course of events. So once, for example, 10 molecules of growth of a growth factor, for example, EGF is an epidermal growth factor, which helps in the growth and division of the epithelial cells. For example, the cells of skin, the cells of lungs, the cells of colon. So under normal conditions, 10 molecules of EGF are required for the growth of an epithelial cells. Once these 10 molecules are produced, they bind to the receptors on these cells and help in their growth. So this is the normal growth. Fine?
Yes, sir. Yes.
Yes, sir.
Are you able to understand me?
Yes, sir.
Okay. So these 10 molecules under normal conditions help a particular epithelial cell to divide. Now, what happens in the cancer process? What happens in a carcinogenic process? Instead of these 10 molecules, 500 molecules are produced. Fine. So when 500 molecules are produced, this particular epithelial cell, which was normal so far, it now divides without any control. It divides to many generations, or it divides very fast because of the presence of additional 490 molecules of epidermal growth factor. So this is one of the example. Once these additional 490 molecules of the growth factor, such as epidermal growth factor, are present, it helps in the abnormal proliferation, it helps the abnormal division, it helps in the abnormal growth of those epithelial cells. And finally, they form a tumor, which goes on dividing, and goes on dividing. So this was one of the example how cancer cells sustain proliferative signal. They produce many molecules above the normal threshold, which helps in their growth. So this was the one hallmark, or the first hallmark, known as sustained proliferative signaling. Another is evading growth suppressors. Another hallmark is evading growth suppressors. Again, I will be able to make you understand by making contrast, by making the differences between the normal tissue or the normal cells and the abnormal cells or the abnormal cancer cells. So as I gave you the first example, that is the cells produce these growth factors which help them to grow and divide. But again, the same cells or their neighboring counterparts have the ability to produce growth suppressors, which help them to suppress abnormal proliferation. For example, Almighty has developed a system within us. He has developed the physiology within us, which is quite ready to overcome any sort of defect which might occur. Say if more of a certain growth factor is produced, there is always the production of the inhibitor of that growth factor. Anytime defect might develop in the cells, any time defect might develop in the body, but Almighty has always kept the safe routes. He has always kept the alternatives. He has always kept the inhibitors which overcome the effect of the defects which might occur over the period of time, or which might occur over the normal course of events that occur within the cells of the body. So I mean to say, even if more molecules of a growth factor are produced, but there are the systems or there are the inhibitors which might overcome their effect. So this is the normal. This happens in the normal conditions. If under normal conditions, 10 molecules, instead of 10 molecules, 20 molecules are produced, there is always a system which can overcome the effect of 10 molecules or 10 additional molecules. Fine. Am I able to make you understand?
Yes, sir.
Okay. Let's move on. So, but the cancer cells are very clever. They have the ability to inhibit those growth suppressors. Under normal conditions, we have the production of these growth suppressors. But cancer cells, because of the mutations within their genome, because of the changes within their genome, they inhibit the expression or they inhibit the production of these growth suppressors, which would have countered the effect of additional molecules of the growth promoters. So the second hallmark was evading the growth suppressors. The third hallmark is resisting cell death. So, and again, I will be able to make you understand by making the differences between the normal cells and the tumor cells. Under normal conditions, what happens is that if there occurs certain change or certain damage or certain mutations in the genome, which might result in a different protein, which might result in the expression of an abnormal protein. So what happens under those conditions, if the cell detects that within itself there is a damage in the DNA or there is mutation within its genes, which might result in the expression of the protein which is dangerous to the cell, or which might result in the abnormal proliferation of the cell, what happens under those conditions is that these cells undergo cell death. So they prefer to die rather than proliferate abnormally. They prefer to die rather than proliferate, rather than grow abnormally, or rather than divide abnormally. So this process is known as, anybody knows what's that process known as?
Apoptosis.
Oh, very good, very good. So this process is known as apoptosis. So under normal condition, because of certain damages, because of the change in the genome of a cell, that cell undergoes process of apoptosis. That is the death of the cell. There is no requirement of abnormal cells in the body. So Almighty has kept in a system a process known as apoptosis, which results in the elimination of the abnormal cells within the body. Now, why doesn't this process occur, or why doesn't this process becomes operational when cancer develops, or when the abnormal cells divide, or when the abnormal cells grow? Why doesn't that occur? So is there any answer to this question? The answer is yes. There occurs the process of apoptosis, but the cancer cells or these abnormal cells again have the ability to resist this process known as apoptosis. So this third hallmark of the cell, cancer cell or the cancer cells, is known as resisting cell death. Now, how does this result? How do they inhibit the process of apoptosis? The answer is by expression. These cancer cells express certain proteins known as anti-apoptotic proteins. Another reason is these cells inhibit the expression of the pro-apoptotic protein. I mean to say, first, they overexpress those proteins which are anti-apoptotic, which oppose apoptosis. And they also decrease the expression of those proteins which are pro-apoptotic, which help in the apoptosis. So this inhibition in the expression of the pro-apoptotic proteins and overexpression of anti-apoptotic protein results in the inhibition of the process of apoptosis, or the process of clean cell death in case of cancer cells. So these cancer cells, resisting the process of apoptosis, ultimately grow, divide, and proliferate very abnormally, resulting in the growth of a full-blown tumor. So this was the third hallmark of cancer cells, that is resisting the cell death. So can you tell me what are the three hallmarks of the cancer cells?
Sir, पहला है sustained proliferated signaling.
Right. Second one?
Sir, दूसरा है, second one is evading growth suppressors.
And the third one is?
Resisting
cell death or resisting apoptosis. Fine, fine.
Apoptosis, yes, sir.
Okay. Everybody there, or it's just a formality? Are other people there?
No, sir, I'm sorry.
Hello?
Yes, sir, we are.
Okay.
Yes, sir, I'm sorry.
Okay. So let's move to the another hallmark that is known as enabling replicative motility. Fine. Enabling replicative motility. Again, let's understand it in terms of differentiating between the normal tissue or the normal cells and these abnormal cells. Have you people heard about an enzyme known as telomerase or the telomers? Anybody?
Yes,
What are
sir.
those?
Yes, sir.
What are those?
Sir, telomeres replicate the telomeres ends of the
Okay.
chromosomes. सर ये टीलोमेरेज सर एंजाइम सर ये टीलोमर की रेप्लिकेशन करता है।
हाँ जी ठीक।
क्योंकि सर वो नॉर्मल रेप्लिकेशन में सर रह जाता है, तो सर फिर इनका टीलोमेरेज काम करता है वहां पे, तो रेप्लिकेट करता है इन एंड्स को, क्योंकि अगर ये रेप्लिकेट नहीं होंगे तो डीएनए शॉर्ट होता जाएगा और अल्टीमेटली इट विल कोलैप्स।
सर
हाँ,
और भी देवरी मतलब डिवीजन सर जो टीलोमेर वो शॉर्टन होती है देवरी सेल डिवीजन।
Okay, but this happens in what? Normal cells. Okay.
Yes, sir.
So basically, telomers are the ends of the chromosomes. They protect the ends of the chromosomes in the cells. But what happens that with the time, as the cells divide, as the cells age, there occurs the loss of these telomeres. Fine. There occurs the loss of these telomers, and ultimately, say if a cell divides to the third generation, I mean to say if a cell divides into the 3 cells, that third cell will receive the less number or the less part of these telomers. But there is an enzyme within the cell known as telomerase, which sort of builds up, which sort of helps in the addition of it helps to make these telomeres and helps the cell or these chromosomes to remain intact. If now there occurs the loss of these telomeres or the telomerase enzyme, ultimately the cell will lose these ends, these protective ends, and the chromosomes, all of the chromosome within the cell will fuse to itself, which will result in the cell death, or which will result in the collapsing of the entire machinery of the cell. Now, talking about the normal conditions, what happens in the normal cells? In normal cells, there is a limited supply of the limited amount of the enzyme telomerase. So it is only able to contribute certain amounts of the telomeric ends. It can only build the telomeric ends to certain point of time. After that point of the time, this expression or the amount of this telomerase is lost, and finally the cells die down. That is known as the mortality of the cells or the senescence in cells. Are you able to get me?
Yes, sir.
Okay. So, I mean to say, a normal cell is destined to die. Even if it divides to certain generations, after those generations, those cells die because of the loss of the telomerase enzyme, and in turn the loss of the telomeric ends. This was the story about the normal cells. But what happens in the tumor cells? Tumor cells basically are immortal. How are they immortal? Because they have the continued expression of this enzyme known as telomerase, which goes on adding these telomeric ends. So even if a cancer cell divides to the 20 generations, it is able to contribute to the number of cells within a tumor. I mean to say, due to the continued presence of the telomerase, the tumor cell is immortal. It continues on dividing without undergoing senescence, because it has the continued supply of telomerase, which in turn goes on adding the telomeric ends, and these chromosomes within the cancer cells remain completely safe without the fusion, and finally they help the cell to attain the replicative motility and the continued division. So this was the third hallmark of the cancer cells, known as enabling a replicative motility. Fine. Were you able to understand this hallmark or not? Shall I repeat?
Sir, repeat.
Okay, repeat. Okay, I will repeat. So I will put it in a different perspective now. Under normal conditions or in the normal cells, we have limited amount of the enzyme known as telomerase. Let's first understand the function of this telomerase. This telomerase adds certain nucleotides to the ends of the chromosome because, as we know, we have chromosomes or the DNA which run in the 5' and 3' direction, and 3' and the 5' direction, because we have two helix present in the double helix. Fine.
Yes, sir.
How many chromosomes does a normal cell has?
46.
Present in how many pairs?
It's 23.
23 pairs. 23 pairs. Right. So are these chromosomes, are these 23 pairs separate, or they are joined together? Are they diffused, or are they are joined together within the nucleus of a cell?
Sir, they are in different forms, whether they may be in interphase chromosome, whether they may be in mitotic form, they may be highly condensed.
स्टेज पे है हम देख रहे हैं.
Like,
Right.
for example, I'm talking about the interphase chromosome.
Sir, interphase chromosomes will be highly condensed.
They will be highly condensed.
Condensed.
Okay, will they be joined together or they are separate?
So joined, but at metaphase we can see them totally alive.
Totally. So this depends on the type of the cell phase. But if we broadly talk about these chromosomes, they are never fused. But we can just see them diffused or separate in the metaphase stage. I just mean to say that these chromosomes are apart from one another. How are they apart from one another? Because they at their ends there are protections. There are protections, or there are ends. Those ends are known as the telomeres. These are the repetitive sequences, 80, 80, 80, having high amounts of 80. So these repetitive sequences at the both ends of the chromosomes confer protection to these chromosomes. They help them to remain intact. They help prevention of the fusion of these chromosomal ends. Fine.
Yes, sir.
Which enzyme
Yes.
helps in the contribution, or which enzyme helps in the addition of these telomeric ends? No, that enzyme is known as
Telomerase.
telomerase, right? So in normal cells, there is the limited supply or the limited amount or the limited concentration of this telomerase enzyme. When these cells divide, for example, a cell having these telomers, it divides into another cell, that divides into another cell. So finally these telomeric ends are lost because the expression of this telomerase enzyme is very limited. Fine.
Yes, sir.
Okay, now when there occurs the loss of these telomeric ends, because of the less expression or the limited expression of this telomerase enzyme, ultimately what happens, the chromosomal fusion occurs, or the chromosomal degradation can occur because of the less amount or the least presence of the telomeric ends or the protective ends. So this happens only in the normal cells or under the normal conditions, and this results in the process known as cell senescence. I mean to say, the cells will only divide up to certain point of time because of the limited expression of the telomerase enzyme and the limited amount of the presence of these protective telomeric ends. And finally, the cells which has divided, which over a period of time they will undergo cell death or they will undergo cell senescence. But what happens in the cancer cells? In cancer cells, there is continued expression of this enzyme known as telomerase, or the overexpression of the telomerase, which results in the continued addition of the telomeric ends to the ends of the chromosomes, and hence conferring the protection to these chromosomes. Ultimately, what happens, this cancer cell does not attain or does not undergo the process of cell senescence, and in turn it becomes immortal, or it attains the replicative immortality. So I mean to say, these cancer cells never undergo the cell senescence, or in other words, they attain the process known as replicative mortality. Fine.
Yes, sir.
Okay, this is the fourth hallmark known as the enabling replicative mortality, or the presence of replicative mortality in the cancer cells, which is not present there in the normal cells. Now, there is one more hallmark of these cancer cells that is known as reprogramming energy metabolism. Can you people tell me the entire course of events that occurs from glycolysis to the ATP production? Anybody among you?
So first glycolysis, then the Krebs cycle, then ATC, and then we get the ATP at different steps.
Well, so if you start with the say glucose molecule, what happens? Glucose
So
undergoes.
pyruvate is formed
Pyru-
at the end.
Okay, then it channels to
Krebs cycle.
the Krebs cycle,
where is pyruvate formed? Why does glycolysis occur? Cytoplasm, or
cytoplasm,
cytoplasm?
cytoplasm.
Cyto.
Okay, so then pyruvate moves inside
Mitochondria.
mitochondria,
Sir.
and then what happens within the mitochondria?
Pyruvate is enzyme that goes into the cycle.
Okay, and finally there occurs the production of ATP.
Acetyl coenzyme.
Okay, so finally what happens is there occurs the production of ATP through the oxidative phosphorylation. Okay, fine. So this is the normal course of event that
Yes, sir.
happens in the normal cells. Starting from glucose enters a cell through the glucose transporters, and it is channeled to the glycolysis. Through glycolysis, pyruvate is produced, and it finally moves inside the mitochondria, undergoes Krebs cycle, and finally oxidative phosphorylation occurs, and we have the production of the ATP. Now, what happens under anaerobic conditions? Do you know about the anaerobic conditions? Say, suppose somebody, what happens?
Yes, sir. Sir, only forget it goes under formation.
Okay, Sahila, can you tell me
Not
what
really.
happens?
So usually the lactic acid is formed in case of anaerobic respiration.
Why doesn't Krebs cycle occur under those conditions?
क्योंकि ऑक्सीजन ही
Bacteria, these organisms
क्योंकि ऑक्सीजन नहीं होता है. Okay, So we have the production of lactate under anaerobic conditions. Am I right?
Yes, sir.
But is this process efficient? लैक्टिक एसिड प्रोडक्शन में कितने एटीपी जनरेट होते हैं और
No, sister, the number of ATP is less in case of anaerobic respiration.
right, and in case of complete aerobic respiration, how many ATPs are produced from a glucose molecule?
6.
38.
Okay, 36 or 38 depending upon the shunt, NADPH or okay. But
Yes, sir. Hexose monophosphate shunt pathway. Sir, that depends on different pathways.
right. Good. But now, can we say that the lactic acid production is the inefficient process?
Yes, sir, inefficient in comparison to aerobic.
Okay. But does that save a cell under those anaerobic conditions?
Yes, sir.
Yes, sir.
Yeah, it does save a cell, because under those conditions we have to generate the ATP. Even if the process is inefficient, but ATP gets generated, which provides the energy for the physiology of a cell, even if the process is very inefficient. Am I right?
Yes,
Yes,
sir.
sir.
Sir, if the organism is multicellular, then what is it?
Ayaz, I am not able to hear you.
If the organism is multicellular, is it? It's only to level.
Sorry. I am your voice comes much
So I
distorted to
think
me.
he's asking that in case of multicellular organisms, is anaerobic respiration, does it occur? I think he's asking this question.
Oh, if he is asking, then the simple answer is yes. Because if you run, for example, uh, if you run a kilometer, or if you run, say 500 meters, after a certain point of time, you need the continuous supply of the oxygen. Your muscles in your legs, or if you perform certain vigorous exercise, they are not able to get that much of oxygen supply which is required under normal conditions. So under those conditions, the process of lactic acid development, or the lactic acid production happens, and ultimately it results in the production of those two ATP molecules, which save your muscle cells, which are helpful or which are required by your muscle cells for their normal function under those vigorous conditions, or under those extreme conditions. Fine.
Yes, sir.
Okay. So a similar kind of process occurs in the cancer cells. This process is known as reprogramming of the energy metabolism. Cancer cells prefer the formation of the lactic acid rather than preferring the complete generation of the ATP or the energy through the TCA cycle. They prefer this inefficient formation of the lactic acid rather than the entire course of events leading to the oxidative phosphorylation and finally the energy production. Can anybody tell me why? Because I have already given you the answer. Why do they prefer this lactic acid formation for the energy production? Sahila, Almina, Majid. Hello? Any answers?
Maybe as cell-saving mechanism to save
No.
it, maybe in this either way.
No, because in cancer cells there is continued supply of oxygen, because they are similar to the normal cells. There is continued. This is not a S condition similar to the exercise where you have limited oxygen. These cancer cells are present within a normal tissue. You have normal cells within them. You have you have normal cells surrounding them. So if the normal cells undergo the complete sequence of events, why do these cancer cells prefer the lactic acid formation?
Maybe because इनको energy उतनी जरूरत नहीं होती है.
They require much more energy. How would they proliferate very much? How would they proliferate to that extent? Then they required much more energy. The answer lies in that.
So maybe because there are large number of cells, they
So
have
what?
to proliferate.
If oxygen for respiration, it needs oxygen all the time. Oxygen not so cells can
Sir, energy को
The
conserve
answer
करने
is
की.
very simple. As I told you that this lactic acid production results in the only 2 ATPs, but the process is very fast, isn't? So the cancer cells require very fast energy production. So that's why they
Yes, sir.
undergo the process of this lactic acid development. Now, if they proceed with a normal course of event, it will take them longer time to produce ATP. Instead, they prefer this formation of lactic acid, even if that produces very small number of ATPs, but those ATP they that help them to drive the processes for their abnormal proliferation. Now, again, they are much more clever. Even if they produce only 2 ATPs, how would they encounter this inefficient process? Any idea? How would they make this process very efficient? Even if it produces 2 ATPs, how would they make this efficient?
So maybe by the passive process of this fission, binary fission.
No, that's not the answer. So they increase the expression of the glucose transporters on their surfaces. Once more glucose transporters are present within their surfaces, more and more glucose is brought in. More and more glucose is brought in, and that more and more glucose is converted into the lactic acid. So if you have, under normal conditions, you have only 10 glucose molecules. Those 10 glucose molecules being channeled through the TCS cycle and finally resulting in the production of, say, 100 molecules or 200 molecules of the ATP. But under this condition, under the production of the lactic acid, even if the process is very inefficient, because only 2 ATPs are produced, but these cancer cells incorporate more and more glucose in comparison to the normal cells. So if under normal conditions you have only 10 molecules of glucose present for the function, for the glycolysis and TCA cycle, but under abnormal conditions or in the cancer condition, you have 1000 molecules of glucose present in these cancerous cells, so that they are converted in the lactic acid. Am I clear? Are you able to understand me what I'm trying to say?
Yes, sir.
So this is the hallmark of the cancer cells. Do you know about the PET scan? Do you know about the PET scan?
Yes, sir.
Okay. So in the PET scan, a dye is injected which detects glucose. I mean to say, once that dye is injected in the patient, it will go to the cells. It will move into the cells and bind to the glucose. In normal cells, you will have very less glucose, but in cancer cells, you will have
More glucose.
Why?
So
Because more glucose transporters are there.
energy.
Process is the production of, the process of production of lactic acid only. So once that dye binds more to the glucose in the cancer cells and very less to the normal cells, it helps in the detection of the cancer mass within a particular organ or within the entire body once a PET scan is done to screen a patient. So you will get contrast images showing more distribution of the glucose to a particular tissue or to a particular place in the entire patient or the entire body of the patient in comparison to the normal counterparts. So in normal cells, you will have very less distribution of glucose, and in the abnormal cells or in the cancer cells, you will have much more distribution of the glucose, so that radiologist says, yes, this is the position or this is the part of the body where there is much more distribution of the glucose, there is much more incorporation of the glucose, and he gives clues, yes, it is the tumor where the surgeon now needs to check for the recession or for the excision of that tumor. So it gives an indication. Yes, this is the part of the body where much more glucose is present, where there is abnormal distribution of the glucose, and hence this part is the part where we may have a tumor. So then it's the responsibility of the surgeon to check for other processes, and finally that a tumor, the tumor in that patient may be diagnosed. Am I clear? Are you able to understand me?
Yes, sir.
Everybody?
Yes, sir.
So this was the fifth hallmark of cancer, known as reprogramming of the energy metabolism. I mean to say, the cancer cells prefer the formation of the production of the lactic acid, even if the process is completely inefficient, rather than following the sequence of glycolysis, TCA cycle, and oxidative phosphorylation for energy production. The reasons I gave you, and I think you have better understood the reasons for this abnormal escape from the normal metabolic route. Clear?
Yes, sir.
So this was something about the cancers, formation of the cancers, reason behind the genesis of the cancer formation, and hallmarks of the cancer. What are different hallmarks of the cancer? What are the properties of the cancer? And how would a person or you people able to differentiate between a normal cell and the cancerous cell? So are these things very clear to you, whatever I talked about so far, or are these things clear to you? Were you able to understand me?
Yes, sir.
Okay. So I think, when do you have another class for the same course?
So tomorrow.
It's tomorrow.
What time?
So at 3 p.m.
So I think I will talk to Raeesar, and I will talk about tomorrow. I will talk about the oncogenes and the tumor suppressor genes. So once we are able to understand what oncogenes are, what tumor suppressor genes are, our understanding of the process of cancer formation or the cancer biology will become much more better, and we will be able to differentiate between the normal cell and the cancer cell, and what happens at the molecular or the genetic level that results or that drives the process of carcinogenesis. Is that clear to you if we talk about those things tomorrow?
Yes, sir.
Yes, sir.
So we will be joining tomorrow the same time at 3 p.m. Fine.
Okay, sir.
Okay, sir.
Okay. Then see you tomorrow. Assalamu alaikum.